A very good afternoon, everyone. I'm Aisha, and I'll be your host for today's webinar. Kindly note that this webinar will be recorded. It is my pleasure to welcome you to our ACTRIS Cell Therapy Lecture Series. Today's lecture by our distinguished speaker, Associate Prof. Tu Heng Phon, focuses on non-viral reprogrammed mesenchymal stem cells in oncology. Associate Prof. Tu is a faculty in the Department of Biochemistry and Director of Research, Yong Lulin School of Medicine, NUS. He was a scientific advisor to the Biotransformation Innovation Platform and a scientist in the Bioprocess Technological Institute, A*STAR. He was also a fellow of the Singapore-MIT Alliance in the Molecular Engineering of Biological and Chemical Systems program and co-chaired the Chemical and Pharmaceutical Engineering program. His laboratory was funded by Roche Diagnostics and the National Institute of Health USA to develop diagnostic assays for infectious diseases. Recently, his team developed a non-viral gene delivery technology to modify mesenchymal stem cells for oncology and regenerative medicine. There are intellectual property protections of specific diagnostic and biotechnology platforms with various agencies and with MIT USA. Now, before we get started, if you have any questions during the presentation, please type them into the Q&A window. Associate Prof. Tu will address these questions after the presentation. And now, without further ado, let's welcome our distinguished speaker, Associate Prof. Tu, please.
Thank you. Good afternoon, Singapore. So what I'm going to do today is to share our work over the years on how we reprogram mesenchymal stem cells. In this case, for today, I'll just talk about how we use it in oncology. Now, the use of mesenchymal stem cells in oncology is emerging, as you can see over here. And you can see clinical trials have been completed and some are recruiting, and there are many more in the pipeline coming into play. So what I'd like to do today is as follows. I'll start with sharing our results on companion animals. That means these are not animals that, like mice, where we put a tumor in. This is a naturally occurring cancer. And we've got some results from that. And we also have first-in-man treatment. Then I'm going to explain how we do it, how it works. And then I'm going to show why MSC is suitable for delivering therapeutics to cancer cells. And I'm going to talk about how we do it in reprogramming it using the method that we have developed and where are we heading for towards the end. Now let's start with results first. Let me show you what we have done. So using this platform technology, we can actually have very high efficient transfection of genes into MSC cells. And this is a platform technology that we can put almost anything we want inside. And it will be expressed at high payload, I'll show you. And we've done it with DNA, RNA, and all the nucleic acids that we can put our hands on. And today, we have these therapeutic cells that have been modified. And we're industrializing it now. And we're going for GMP production of these cells for clinical use. So today, I'll just focus on oncology. Here are some of the results that we have to date. So we've been treating, this is not experiment, this is not a study, this is the treatment for what we call naturally occurring end stage. So these companion animals, these are dogs and cats, they have no more option. They've tried everything, radiation and chemical and everything else, and they come to us for help the last few years. And we have treated more than 65 patients, and it's still ongoing. We have done peritumoral, that means we put around where the tumor is, and we also have done systemic, that means we put through the bloodstream. And to date, what we like to say is that it is safe. We have not seen a single case where the companion animal have any severe adverse reaction. And we have seen, to our surprise, There's some of them extended life, and I'll show you some of those. 1 year of dog's life is about 2 to 5 years, or some people say it's about 7 years of human life. And we have some of these companion animals that live more than 3 years. And it's very easy to use. You put the cells inside your syringe, and all you have to do is inject to the site or into systemic, and then you take a pill, and that's about it. And I explain how these things work. So first question is, why do we want to treat companion animals? If you look at dogs, the main killer, the first killer is actually cancer. And it has been shown by NCI that if you treat dogs, you're learning from them and you'll actually be able to help humans as well. Genetically, they're very similar to us. So that's why whatever you do in dogs will be useful in human. And because of this, what we've been doing last few years, we checked with FDA. There's a new program called the Veterinary Innovative Program, and we have opened a master file with US FDA so that our treatment one day can go to human. All right. Here are some examples that this is peer reviewed since last year been published. Now you look at this particular tumor over here, it's called perianal, it's around the anus area. And when you cut it out, in this case, it kept recurring, it kept coming back. And there was nothing much the surgeon can do or the vet can do. And after the treatment, you can see the fur recurs. And this animal or this patient have lived for more than 30 months. It died of frailty, that means getting old age, died of old age beginning of this year. Here is another case where we injected into the bloodstream systemically, and we see a shadow before the treatment in September 2020. And over the years, you can see the shadow is still there, but this dog do not have any of the phenotype anymore. Like they are very stable in disease and do not cough at all. This dog is still at home, is very old, is still at home. Now, these are two examples, but beyond that, we have others. Like here, we have a recurring sarcoma. This was one of the first cases we treated in 2018. And what you see here is a big gaping hole, simply because they keep excavating the tumor and it keep recurring. And after the treatment, you can see the wound heal, and then it lived for more than 32 months, and it died of old age, frailty. Here's another one. You can see they keep cutting to the bone, and the tumor keep recurring. And after treatment, you can see it's gone. Here is another one, and then you can see the fur recurs. Here is our collaborator in Thailand. You can see the big bulk over here. Then after the treatment, the disease is basically subdued. And here's a collaborator in Hong Kong. We were doing a clinical trial with them, and you can see a differential sarcoma, and after the treatment, it's collapsed and it's gone. So these are very promising results. And it's more than 15 months now for this case, there has no recurrence. A quick warning, but I'm not going to show all the detail. This is a first in human. So I gave a talk in Indonesia sometime in September 2019, and some clinician came up to me, and we have a case. They came to us for help. So November 2019, what happened was that this patient is essentially has no more option, and it was very bad. They suffer from what we call a clear cell carcinoma. When you have this, it's almost nothing we can do. They have tried debulking the tumor. They have radiated her, chemotherapy, nothing worked. I didn't want to show in detail, it's very ugly. These are two legs, and essentially it's eating out of her from the abdomen, from the ovary region, invaded the whole area, and keep eating her alive. And after our treatment, we contained the tumor. This is a WhatsApp from the clinician over there. This is in Indonesian, so it needed to be translated by them. They call this treatment Treffen. And after the first treatment, he said the pain at the wound where this pain is, you can see is bulging out here. He says no more pain. The area around the wound has softened. You can see a grayish area. The missus and husband are really happy. After the first treatment, there was no shivering, no side effects. So over a couple of months, what we have observed is there's a collapse of tumor bulk, and she's no longer anemic, and she has improved in appetite and pain-free. So the quality of life has improved. The sad part of this story was that in 2020 February, together with lead scientist Dr. Ho, we flew over to Indonesia to talk to one of the 4 clinicians. And our idea is to now chase the real problem, which is inside her. We didn't have a chance to actually perfuse the cells inside her peritoneum because of COVID. So we came back and there was a lockdown. We had to throw everything away, and they had to throw them away. And the clinician visited us about a month ago, came to Singapore, visit us. We're going to restart some of these trials. And she told us that she was still alive a couple of months ago, but developed secondary tumor. So I wish we can do better. So because of all these things, we ask ourselves the question, whether we can actually move forward with this. So the data seems very promising. How does it work? So essentially what we have done is we implant or we transfect a gene inside. It's a yeast gene. This gene actually is found in fungus, and you take a drug called 5-fluorocytosine you can buy for your fungal treatment. And with this gene, it will be converted into 5-fluorouracil. This is well known to be a toxic drug for dosing oncology. So in colorectal cancer, there's a regime called Frolox. They use 5-FU very frequently. And the nice thing about this, I'll show you data afterwards, MSC can actually, they sort of like crawl their way towards a tumor area. And then over this side, when we switch it on, it essentially 5-fluorouracil kill off the cancers and releases the guts of it, releasing neoantigens, which in turn inform the immune system through, later I'll talk about, the C-gasting pathway. And here, the 5-fluorouracil will corrupt the DNA and RNA biosynthesis. Now, if you were to use 5-fluorouracil alone to treat, you find a lot of systemic issue. 5-fluorouracil is good, it's just that it's very toxic. And you can see over this side, it's published with somebody else, where you use MSC, you carry the gene, you switch it on, there is no systemic issue. Now, how does this part work? Essentially, it's published by a group in Yale. They published in EMBO Journal about a year and a half ago. And what they show is 5-fluorouracil, when it gets into the cancer cell, it basically breaks it down and kick off the C-gasting pathway, releasing interferon beta, which brings in the T cells. That explains why, in our case, the recurring cancers in some of these companion animals do not recur after the treatment. So this is a very elegant piece of work by the Yale group. So what we do is essentially have 2 arms, a chemo arm, which basically releases it on the spot, and the immune arm that brings in the immune system. Here are a bunch of results that have been published. We are focusing on glioblastoma. You can see the cells, resistant cells kill equally well when you use mesenchymal stem cells to carry the gene and when you switch it on. Here is using a bunch of cells from Uppsala, and this is actually cell lines which are very well characterized and reflects the tumor in the human. And similarly, you will kill them off. Here you can see in vivo, you can see what happens over here. If you treat it 3 times, if you don't treat it, it continues to grow. Here we have the neurosphere from human patient. Recently, we developed that with Tan Boon. And you see the MSC comes along. Without switching it on, the tumor keep growing. And when you switch it on, they dismember every one of them. And this tumor is actually in the dish, is TMZ resistant, which is the first line of drug. And you can see even when you switch it on, without the stem cells, nothing happens. Now, so we have a hypothesis that if we use these engineered cells to deliver this kind of chemotherapeutics, it will be useful. Why? We think this way is this. In the tumors region, you can see it's infiltrative, hypoxic environment, and it penetrates. Basically, they spread themselves out. and the biology is not easily to be whacked by CAR T. And this whole area is basically a cold tumor. So what we found was that MSC is great because they love to stay around this area. And even it's hypoxic, they're fine, they live there well. And they nest or home towards this tumor, and they can migrate naturally. And all we have to do is program it. We can program it with different kind of method that you've seen just now. Using the chemoimmunotherapeutics, we can actually program the cells to carry the therapy towards it. Essentially, what we're doing is creating a Trojan horse. So this will get very close to the tumor, switch it on, and let it kill, and then bring in the immune system. Now, why MSC is a good tool for delivering drugs is not necessarily a new thing. First of all, there are more than 1,000 clinical trials going on using MSC. They're safe. Very important, they're safe. And you look at the trials, nowadays, you can see UCL all the way down. They have now put, using MSC, they can even put viruses inside to carry it to the site that they need it to be. Essentially, they are very desirable. They have low risk of rejection, and they have this homing capability. And I'll show you more data on this. Now, the tumor microenvironment is a natural habitat for stem cells. They love to go towards where the tumor is. Now, this was published by somebody else. You can see the MSC actually migrate greenish color towards where the tumor really is. Here's another one using the neurostem cells. They program it, you can see it moves towards the cancer cells. Here is something we've done using the neurosphere. They form into 3 dimensions. You can see 3 hours after plating, the green color is actually the MSC carrying the gene. And you can see overnight, very spectacular and remarkably, you can see all the MSC basically home and zoom towards where the tumor really is. So this is a great tool to have so that we can transport things towards the tumor. Not a magic. Why do they tune? Why do they nest towards MSC, towards tumor? Because it's been published, well published by a lot of groups now. The tumor releases a lot of factors that basically the receptors on MSC they have, and so they can nest towards the tumor area. Now, here is something we have done. We have shown that in vivo, you can see ES2 actually is a peritoneal cancer, and you can put inside, you can see it develop very well. And when it's inside there, you put the MSC, you can see it stick around where the tumor really is. Now, why do you need to modify MSC? That's very important to note, is that it is well known as well, has been published by other groups, that some of the MSC can promote tumor progression rather than suppressing. Some they can suppress. Now, here's a great study I've shown some time now, that if you put native MSC, certain kind of MSC, you never know what the donor looks like, and it can actually promote it to become bigger than the control. Then you have to kill it using something very similar to what we talked about. And this has been carried out in human for phase 1. And they've shown that even using a viral program, MSC, and you can see it's pretty safe. Reprogrammed MSC is necessary to enhance function. As you can see over here, a lot of publications now begin to recognize the importance of modifying the stem cells to make them more powerful, if you like. Now, here is one study by this company called SendBio with Stanford. They've shown something in regenerative medicine. You modify the MSC, you actually can rescue some of the phenotype when you have a traumatic brain injury. So you can see on these cases here that modifying MSC allows you to increase the function that you need. But then how do you reprogram it, right? How do you transfer things into it? Obviously, everyone knows you can use virus. They're great, but there's significant challenges. Yeah, scalability, cost, you need specialized facility, time consuming, safety, obviously, regulatory hurdle. On this side, non-viral would be great. You have a chemical method. The DNA usually is not very efficient. mRNA is great. You can modify, and I'll show you data afterwards, but they don't stick around too long. They're limited potency. And then on this side, you can do electroporation, ballistics. Generally, you have difficulty in scaling and is not very potent in a way. So over the years, what we did was work out the mechanism. This was published in Nucleic Acid Research some years back. So we worked out that actually things do get in, but you need to reroute them very early on. So we call it rerouters. They can reroute them out of this compartment and stabilizing the microtubule, you can actually get DNA into the nucleus pretty well. And so by doing so, you can see later, it increases the potency because we don't use viruses, it's safe. And because we don't use viruses, regulators are lower. And our product cycle, we can produce one. Every cycle is about 1 week. I can produce enough for 6 patients use. And I'll show you a bit more. And obviously, we don't do all this complicated stuff, then developmental costs will be lower. Now, here is what it looks like using what we have. If you use viruses to modify the stem cells, this is mesenchymal stem cells. These are the same stem cells from rooster. You can see, yes, there are a lot of cells get modified, but you can see the payload is low. You have to really crank up the background to see it. And over here, using what we do, you can see very high payload. Then we did in parallel with mRNA as well. With the same exposure, you can see the vast difference between mRNA program and DNA program. You have to tune it up before you can see it. This is effects data you can see, whereas over here is always to your right side. The nice thing about using this DNA is that now we can do what we call cellular object-oriented programming. It's like JavaScript, right? So what we have done is program in such a way that over time, it increases. If you use RNA, it will go up, come down very quickly, and it disappears. So we've done that, and we can see it does not affect the cells at all. We can even transfect primary neuron or even neuronal cells. And you can see after you program it, you can still differentiate very well. We have done metabolic analysis. After you wash it out, the cells are the same. We have done with a whole bunch of other MSC. We now can develop allogenic MSC off the shelf, ready to use. All of this works very well. And obviously, it does not change the MSC characteristics, which is very important. And essentially, what we have done is we have created a hunter-killer, isn't it? Think about it. So what we have done is the MSC can hunt on its own. It can home on its own. And then we basically program it to become a killer. So this is a chemo immunotherapeutic arm. So I'm going to end soon. So what have we done next? We are going to scale up now. We're going upwards. We see an unmet need for rapid industrial relevant scale production of these modified cells. So we have explored, you have seen just now, with different contributors. We can actually take the cells, we go through, and we can check them out, make sure they are good to be transfected. We have now done design transfer into ACTRIS and HSA or CTF. And what we've done now is to expand the cells for GMP production of clinical grade for human use. In the process of doing all these things, this is something maybe basic scientists can learn about as well, is that sometimes when you do R&D, it cannot be translated into large scale or regulated. So in our case, all our processes is what we call design for manufacturability, assembly, and utility. So over the R&D stage, all our processes were designed so that one day we can transfer all of them over to GMP. So that's why we can do things very quickly today. Here is an example. The lead scientist, Dr. Ho, is holding up a billion cell. This is what it looks like. And you can see we do in 2D, one operator. We can do 1 billion cells a week very easily. This is on 2D. We have also done it in 3D using microcarriers. We're not going microcarriers at the moment, not because of the scale-up issue, but what happens here is simply we're waiting for somebody to go ahead of us, get it regulated, get it approved, then we can actually scale using microcarriers. We also now have to think about how are you going to take it away and inject into human one day. So we have developed our own preservative. It's DMSO free. We did it by algorithm-assisted design. We do a lot of math and stuff like this. We found out a certain kind of mixture is great. And these are all clinical grade stuff that we can use. And we can actually see that it does not change the cells after you have cryopreserved. All right, so what do we have today as a whole is this. We can actually take cells, MSC cells. We can actually harvest our own, which I haven't shown you the data. We have done that successfully, but it's not GMP, so we don't use those. We work with third parties to take third parties' GMP cells, and we can go through the whole process. And all these things, we have a total solution from the start to the very end. So that's what we can do today. Now, it's very important that although we started off with transfection and we work out all the mechanism, we have to develop the processes, the media, and everything else, all the way from the start to the end. Otherwise, you'll be stranded. You cannot move forward, and you can't take things forward. So where are we going today is this. We are forming consortiums now. We have a clinical collaborators for GBM. Professor Singh Sir from NUH is working with us. And we have assembled a team for brain tumor studies. And then with William Huang from SGH and Johnny Ong. And we have worked with them on Gen 0 and Gen 1. And you can see here, this is their work. And you can see that you can actually kill off or cytoreduce all the cells. Here, for example, you can see untreated. These are treated mice, you can see, versus untreated. This is a human form, ascites form when you have an ovarian cancer. So these things are promising, and we thought maybe this is something would be useful for human. So by end of this year, we should be able to use it for humans soon. So summarizing what we have done is as follows. I've demonstrated the use of a non-viral, scalable, very facile way of delivering genes to the MSC. And if you work in MSC area, you realize it's not easy to transfect cells. MSC, and I have shown to you, is a promising vehicle, and it can deliver therapeutic agents. And we have developed the next generation modified cells to enhance the pathway I was talking about, the C-gasting interferon beta pathway. And right now, we are work in progress. We are now going towards validation stage of a GMP production. My lab is very small. It is what I call the ludicrous team. I want to go 4 guys to do all this, including submission of IND. But we thank everybody, including ACTRIS and CTF, for actually helping us to do this. Acknowledgement, these are the 4 that we have. And funding, this is an example of how a village came by to help what we do. And our medical school has been really, really supportive of what we do, and we're very grateful. Thank you.
Thank you, Prof. Tu, for a very elaborate presentation on your work. We will now proceed with the Q&A session. So we have one question in that. This question is from Aditya Narayanan. The question is, have you tried bioencapsulation of the MSC with alginate or similar biomaterials?
Yes, we have not tried alginate. We bought the alginate 2 years ago because we thought of encapsulating it to give them more release control, but it was not necessary, as you can see. The difficulty of that is that we have to go to GMP production. There'll be more questions coming out, right? So make things as simple as you can. We have done it encapsulating in collagen. We have done it encapsulated in the clinical grade tissue. So all those things do work well. It's just that right now we don't need to use it. Yes, we have done it.
We have one more question from Chelsea Wang. The question is, could you share more details regarding the algorithm that helped you to come up with the DMSO-free cryopreservation medium?
Okay, I can't reveal too much at the moment because we are submitting the manuscript. That's why some things are blanco, just to show you it's possible. You can do it by yourself. Well, there are various ways of doing it. You see, I've got a company called Merexis as well, right? So in Merexis, we do a lot of AI as well. You can also use AI for this. You can just do design experiment. You just have to know what parameters to tweak, and that will help you along with it. Can it be applied to other cell types? Yes, we've done it with neuron. We've done it with, well, I would say that we focus on adherent cells, right? We are not diverting our strength towards CAR T or so. We've been asked many times, can we help with CAR T? We don't have experience in that because I'm not going that way. To me, CAR T is good for hematological cancers and is well known, and recent clinical failures of CAR T in solid tumors. That's why we were building up last few years, addressing the solid tumors. So we don't have experience in those suspension cells. But after saying that, we did do some suspension cells and they do translate well.
Right. If anyone else has any questions, please type your question in the Q&A function and we'll take your question accordingly. Alternatively, you may click the raise hand button, unmute yourself and proceed to ask question. So we have another question. It's not a question. Chelsea says, thank you very much, Prof.
Thank you.
Any more questions, anyone? At this point of time, I don't see any further questions from the audience. So I guess we will wrap up our session for today. Prof, do you have any other interesting facts that you'd like to share to wrap up your presentation?
Not really. Just want to remind everyone that in Singapore, what we're trying to do is build out the cell therapy. And we are just trying to contribute to this national initiative.
We have one more question, Prof. Question is from Jong Yun Han. Do you have an issue with heterogeneity of MSCs, donors, etc.?
That's a very good question. One of the things that very early on, this is more than 10 years ago, we have decided not to use native MSC because most people use native MSC, right? That heterogeneity question is a very significant one. And especially when you go GMP production, you find a lot of variability. So everyone is trying to sort them out based on size or based on properties. Now, I've shown you a slide just now with a science advance and so forth, talking about enhancing the function. The moment we do what we do in programming it, then this issue of heterogeneity is no longer an issue because all of these cells will start doing what I want it to do. That is our thinking. That's why we want to program it to do what we want it to do. Then heterogeneity is no longer an issue.
Ray says, thank you, Prof, for the session. And we have another question from Cyrus. I may have missed this detail, but do the cells stably express the genes?
Very good question. So in our case here, okay, I'll share with you a funny thing that I've been working on for all these years, right? Before COVID, I have a lot of problems. Everyone is asking and saying, oh, you do this GMO, GMO, they're very silly people, right? After COVID, nobody asked me the question. If they are there to ask me the question, I say, are you GMO now, right? Frankly, then of course you have CRISPR. Don't forget about CRISPR. Don't forget about CAR T, which is also programmed, right? So the silliness have gone because we know today when you program, depends on how you program, it can stay on or get off. Now, we deliberately, for many years, did what we do using transient. So you look at our programming, right? It went up And for many days, it can stay up for up to 21 days. In vivo, we have injected the cells and retract them for 21 days. And these guys are still around being modified. Why do transient when we can do all this integrated, right? The integration or transient depends on the vector you use. In our case, we deliberately do not want to integrate. Party integrates. Using lentivirus will integrate. When you integrate, go to FDA website, you will see for yourself. You're only allowed to do 3 to 4 or 1 to 5 copies maximum. Why? When you integrate, you don't know where they're going, right? Maybe by going to the wrong place, you'll cause problem, right? So we have decided some years ago not to do it simply because we thought we're going to have a transient that is equally powerful, if not more powerful. Don't integrate, then it die over time, so you don't have a footprint. We don't leave behind a footprint. That is what we do. So ours is transient, but even a transient, you can see if you program it correctly, it can go on for some time. So would this be considered a therapeutic cancer vaccine? Another good question. We can actually do vaccines very easily. There was a study actually during COVID time from Tsinghua University, published very early on. And what they did was use MSC, put in different parts of COVID virus, inject into the animals. Not surprisingly, they could raise antibodies, robust antibodies, much more robust than mRNA can. And in fact, there is already a patent a couple of years ago in the US when somebody patented using MSC to use it as a vaccine. So there's nothing to stop me from taking the MSC, putting in a new antigen into it, expressing it, and provoking an immune system for cancer vaccine. Nothing to stop me from doing that. In fact, in our lab now, we are developing all sorts of things. We're using MSC as a biofactory. They're just making a lot of different things I wanted to make. So the key is, how do you make them well? That's where we come in. Good question. There's one more question.
Cyrus says, thank you for your amazing work.
Thank you. Thank you.
All right, does anyone else has any other questions? If you do, you may type your question in the Q&A chat, or alternatively, you may click the raise hand function. Okay, we have another question from Shu Uigan. Thanks, Prof. Would you be able to re-administer the cells? Re-administer?
Yes. I don't know whether I'll show it to you. Wait. Here is one. Oh, cannot see well. Cannot see well. Never mind. We have administered over and over again, can be up to 8-9 doses for the human. You have seen earlier 8 doses we have tried where we have injected. Yes, you can re-administer. Why? Is because MSC generally is immunoprivileged. They don't express MHC too well, and the MSC1 is not very highly expressed. So when you keep injecting it, they're not rejected. And we do see in the 65 animals, the companion animals, that we have injected at least 3-4 times, and there is no immunoreaction, there is no rejection of the treatment. Yes, you can.
Right. Anyone else has any questions? If you do, please type your question in the Q&A function and we'll take your question accordingly. Alternatively, you may click on the raise hand button. You can unmute yourself and proceed to ask questions. Right, at this point in time, I don't see any further questions from the audience. So we will wrap up our session for today. Thank you very much, Prof. Tu, for the very interesting, very engaging talk. And to all our audience, thank you for attending today's lecture. Thank you again, everyone. Thank you, Prof.
Thank
Tu.
you.
Thank you, everyone. Have a very good afternoon.
One last question.
No, please, thank you, everyone. Goodbye.
Okay. Thank you. Bye-bye.